Hello, everyone. My name is Stephanie, and today I'm going to be talking about the effects of endocrine disruptors on marine mammals. So why should we care about this? Well, understanding how these endocrine disruptors affect marine mammals is critical for the conservation of marine species, as well as the conservation of the ecosystem that these marine species reside in. Not to mention that any information on these endocrine disruptors could have potential but could be potentially parallel to a system in humans or something like that, and that might help in understanding more about us as well. So what are endocrine disruptors? Endocrine disruptors are chemical substances that can interfere with the endocrine system. They can be found in plastic, pesticides, industrial lubricants, sunscreens, hair products, etc. The ones that are going to be discussed in this presentation are phthalates, bisphenols, polychlorinated biphenyls, PCBs, dioxins, and DDT, which stands for dichlorodyphenyl trichloroethane. So how do these work? These are just simply some mechanisms for the EDC, or the endocrine disruptor chemical. They mimic hormones, they bind, they can either act as an agonist and activate biological response, or they can act as an antagonist and block the biological response. So this following research is going to be talking about the immunological effects of high PCB concentration. So in this research study, two groups of captive harbor seals were studied. One group was fed a highly contaminated fish diet called the Baltic group because they were fed herring from the Baltic Sea. And one group was fed a low contaminated fish diet. The Atlantic group, they were fed herring from the Atlantic Ocean. 17 blood samples were collected in different time periods. The YA1C tumor cells were used to test for cytotoxicity, and the release of CR51 reflected that natural toxicity. Like I mentioned, blood was taken 17 times to test for immunology, and low cytotoxicity here means immune suppression, and these dark filled-in lines are referring to the Baltic group, so the group that was fed a high contaminated diet. So this means that low cytotoxicity in this group meant that high PCB concentration, or could be correlated with the fact that high PCB concentration could be the reason for this low immune suppression. Correlation does not cause causation, so important to note that. So this next study is going to show high PCB concentrations, PCBs and DDTs, and morphological effects of the skull. So this research looked for skull asymmetry in Baltic gray seals, and they looked for skull asymmetry in Baltic gray seals that lived past the 1960s, so after the 1960s. And they compared it to the skulls of those that lived before the 1940s. Why? In the 1955s, there was a high number of pollution from DDTs and all these endocrine disruptors, right? And that occurred in 1955. So they wanted to see if there was any morphological change because of this high pollution. This table from both of them show A, the pre-pollution sample, the animals born before 1940, and B, the pollution samples, the animals born after 1960. And they show in all characters, oh, I forgot to explain that, this figure shows all the areas of the skull that they looked for asymmetries in. So in all those areas of the skull, in the pollution sample showed high asymmetry. So it did show asymmetry. And so then we can say that there was a correlation between high PCB and high asymmetry in the skull. This following one is high PCB and endocrine effects. Excuse me. So this next study, this was actually a recent study in 2020, which studied 39 plasma samples of ringed seals. Phoca hispida, and 38 of gray seals. So the sex hormones tested here were progesterone, 7-alpha hydroxyprogesterone, testosterone, estradiol, and estrone. And these are just codes for these in the graphs. They had a reference seal group and an exposed seal group. The exposed seals were seals from the Baltic Sea, and the reference seals were seals from Svalbard and Sable Island, which had low PCB concentrations. This graph here shows the mean hormone concentrations of exposed and reference seals. As you can see, the exposed seals had lower hormone concentrations regardless of sex and regardless of species compared to the reference. This figure shows the hormone concentration in relationship to the PCB concentration. You can see in almost all of these some sort of relationship between both, which is good. The progesterone and the PCB concentration have a weak negative correlation, but you can see that as PCB increases, there is a slight decrease in progesterone. Now let's talk about some reproductive effects. This study studied reproductive failures in minks. They had 6 groups of 20 female minks, and each group was fed a different diet of PCB. Group 2 was fed a high diet of PCB. A50 is is Clofen A50, which is a commercial PCB brand. So group 2 was fed a high PCB. They were fed 0.70 milligrams of A50 3 times a week. Group 3 was fed 0.24 milligrams of A50 3 times a week. The A50 compound was then fractioned, and then 0.1 ortho of CBs were fed to the minks in this group, and then 2,4 ortho were fed to the minks in group 5, and then group 6 was fed gray seal blubber extract. And the concentration of PCB and DDT was 0.064 milligrams of PCB and 0.021 milligrams of DDT. On here on your right, you can see this is the weight, the percent weight of the thymus of newborn kids in the first season of reproduction. There were 2 seasons of reproduction for these minks. In the first season, the A50 high was the only one that had a significant decrease in the thymus body weight. In table 5, it shows the reproductive performance of minks after the 18-month exposure. So they had babies, and they tested the reproductive performance of female minks after 6 months. But after 6 months, there wasn't much variation aside from the A50 high that at 6 months had a reduced birth weight, total birth weight of the babies. In 18-month exposure to PCBs, however, A50 high showed a significant decrease in percent mated female whelpings, in litter size of live kits, in litter size and dead and live, so in total liver size, and in birth weight as well. So this shows a high concentration of PCB having a negative effect on reproduction. In A50 high, none of the offsprings survived after 24 hours. They all died after 24 hours. And here you can see the number of offsprings that made it to that 2-week mark. Next up is killer whales. Killer whales are considered one of the most PCB contaminated mammals, and it is predicted that this high contamination is going to have negative effects for the future of these organisms. Because of their high trophic level, they consume a lot of already contaminated organisms, such as seals or seabirds. They eat fish as well, and so all of this builds up. This research showed that that an increase in maternal PCB causes a decrease in calf survival, and it also showed that after a certain concentration of blubber PCB, there was a decrease in immune response. C shows simulations of different areas and the impacts of reproduction and of immunity in blue. Immunity is in blue. And this is the reference population. Okay, so research difficulties. So it's difficult to establish a baseline of EDC levels of all marine mammals. It is difficult to test some of these marine mammals as well. There's also not a lot of recent research on mechanisms of endocrine disruptors. And it's difficult to create experimental research where you can actually grab some of these marine mammals and maybe inject them with something, because that is very unethical. So there's ethical restrictions to this project as well. I hope that this helped understand a little bit more about endocrine receptors. And thank you so much for hearing me out.